19th, and it is, of course, the 6 o'clock class. So we're starting a brand new chapter now, and this is where we really start getting into things and so on. We did the microscopy, we talked about staining and so on, but we'll start doing this week. We'll start doing the gram stain, some of the other stuff we talked about. But right now, I'm actually talking about bacterial cells, so prokaryotic cells. First off, no internal membranes. Now, remember what the organelles are that have internal membranes. The endoplasmic reticulum and the Golgi apparatus. Well, think back on what happens with those organelles. The endoplasmic reticulum, particularly the rough ER, has ribosomes, and those ribosomes make proteins. When the proteins are released from the ribosomes, from the endoplasmic reticulum, the ER actually does some packaging. It will add the sugars to the protein, making them into glycoproteins. But those sugars act as address labels. You know, we did this already. Act as address labels, sending them to the Golgi apparatus. The Golgi apparatus does more labeling and then tells the glycoproteins their final destination, usually exported out of the cell. What I did not tell you before, but I will now, is there are actually two categories of ribosomes. Free ribosomes and bound ribosomes. Free ribosomes and bound ribosomes. The bound ribosomes you've already seen. Those are the ones that are attached to the rough ER. But there's also another category called the free ribosomes. Those are not bound to the ER. Those are just loose in the cytoplasm. Loose in the cytoplasm.
Did they ever become attached to the rough ER?
No, they're loose in the cytoplasm. Now, ribosomes are ribosomes. They make proteins. But the way it works out, the proteins coming from the free ribosomes stay in the cell. They're basically, it's not working. They're basically for domestic consumption, if you will.
You say that again?
Yeah, the free ribosomes produce proteins that stay in the cell. So my comment was for domestic consumption. They stay in the cell. The bound ribosomes would send proteins up to the Golgi apparatus. They make proteins that will be exported out of the cell. So both categories are making proteins. But the free ribosomes make proteins that stay in the cell. The bound ribosomes make proteins that leave the cell. They go somewhere else. So that brings us now back to this business of no internal membranes. And that means no Golgi apparatus, no endoplasmic reticulum. Why would bacterial cells not need Golgi apparatus?
They don't have no
Critical
package.
thinking time.
Well, they don't need it because they're not packaging protein, right?
Well, but the Golgi apparatus can. The Golgi apparatus can package proteins.
And also, is it because they're not part of a larger living, or like they're all
Exactly, they're one cell.
Oh.
I'm rephrasing, but you got the right idea. They're one-celled organisms. There's nowhere to export to.
Right.
For multicellular, you know, us and animals and everybody, for multicellular, well, the cell has places to export to. But a one-celled organism, there is no other place.
So the bacteria has the free.
Exactly. So a bacteria would only have free ribosomes. And those proteins would just stay in the cell. Exactly right. Okay, so no Golgi apparatus because no place to export to, and therefore no ER either because there's no Golgi apparatus to send it to.
Did you say, but they do have ribosomes?
Oh, it definitely has ribosomes because it needs proteins. So it only has free ribosomes, not the bound. It can't be bound because no endoplasmic reticulum. Again, the whole idea is they're unicellular, just one cell, no place to export to.
The bacteria in the cell, right?
Pardon me?
Are we talking about the bacteria or still?
Oh, yeah. Yeah. The bacteria is just a one-celled organism.
That's why it's all ends because of one.
There's no place to export to. There are no other cells.
What about like the ribosomes and mitochondria?
Actually, bacteria don't have mitochondria. And also, mitochondria don't have ribosomes anyhow.
Okay, the wrong medicine.
Yeah, you do. The mitochondria makes energy, does not make proteins.
What about they have their own DNA?
They do. That's not. Also, no histones. So I know you're all thinking, what the hell's that? Besides the fact that you're thinking, who cares? But that's a different issue. So let's go back to the beginning of time, or to the fertilized egg, whichever comes first. You got the fertilized egg, and then it's going to start dividing. This is mitosis. Now, of course, the bacteria don't do mitosis, but we do, everybody else does. So it starts dividing, and up to 7 cell divisions. 7 cell divisions. Which means 128 cells. If you take out any one cell, you get a brand new organism. And the remaining 127 give you a brand new organism. Is this reflecting? Are you having trouble seeing? Let me put that down. And then it starts developing just like the other cells. So you get your identical twins or whatever. Okay. Now at 8 cell divisions, something changes. So that would be 256 cells. 8 cell divisions. Now if you take out a cell, it will not develop into a new organism. And the remaining 255 may develop into an organism, but now you'll be missing something. You'll be missing an arm or a leg or an eyeball or something or other. You'll be missing something. So something has happened. Well, to explain what has happened, let's go up to the present, right now. Lots of cells. Every cell has exactly the same DNA. Well, that's because you're starting off with one cell, it divides, and every time it divides, it's duplicating the DNA as well. So even many cells later, still every single cell of our body has the same DNA. So there's an obvious question. If every cell has the same DNA, why do we have different cells? There's an obvious answer. The obvious answer is that in any one cell, not all the DNA is operating. Some of the DNA is on, and some of the DNA is off. In fact, most of the DNA is off. So depending upon which DNA is working, which DNA is not working, that gives us different cells, skin, muscle, nerves, and all the rest of it. So it depends on which DNA is on, which DNA is off. It's called cell differentiation. Cell differentiation. We get different cells. clearly what's going on is that process is starting here with the 8th division. With 7 divisions, or up to 7 divisions, all the DNA is still operating. It's all still on. And that's why when one cell comes out, all the DNA is working, you get a new organism. But at 8 divisions, some of the DNA has started turning off. So if you take out a cell, not all the DNA is working, you don't get a new organism. By the way, just off the record, off the topic, these first 7 divisions, these are the fetal stem cells we've all heard about. And they're very, very much in demand by scientists because all the DNA is working. So if you take some fetal stem cells, you put them in with brain cells, for example, they'll start developing as brain cells because the DNA is able to do that. But at 8 cell divisions, they can't do it anymore. Yeah.
How long does it take for fertilization?
Can you say that one more time?
Wherever the DNA wherever the histones bind to the DNA turns off. The histones are like off switches. So they have to bind to the DNA first, and then they turn it off. Now, one thing I've never known, so I was hoping nobody would ask me, and thank goodness nobody did, but I'll just tell you anyhow. One thing I've never understood is, how do the histones know where to bind? How do they know which DNA to bind to turn it on or turn it off? Thank you for not asking. So, it's something I've never known. And
Now you wanna know.
too late. You can't ask.
So when you say turn it on, you mean that they just don't bind?
Correct.
Okay.
Yeah,
They
yeah,
don't
it's a good
necessarily-
point. They're already on. So really, they're just off switches. If the histones bind to the DNA, it turns off. If they don't bind, the DNA turns off or stays on. Good point. The DNA turns on.
What causes them to find spots on DNA?
Well, that's what the self-differentiation is. I mean, that is what it is. But again, how do they know where to go? And I do not know that. I do not know that. Let's just say there's still lots of topics of research out there. Because this would be a major thing. Because this would control heredity. Because these histones are knowing where to go. Anyhow, stay tuned. You're not going to find it out this quarter, but maybe down the road somewhere, somebody will figure it out. Okay, back to this. So, bacteria don't have histones. Why not?
No subdivision.
Pardon me?
No subdivision, so there's
No
cell
cell,
separation.
well, yeah. You're right, no cell division. I was thinking of something different, but you're right. No cell division, what I was thinking is no differentiation. All you have is the same thing. All you have is one cell. They're not going to differentiate. So basically, all the DNA that's there, it's going to be used. There's no point turning some DNA off because then why even have it?
So this is
Yes.
all for the eukaryotic.
Go ahead.
This is all for the eukaryotic cells.
Yeah, this thing is for eukaryotic cells. So this is what we do in animals and all the rest of it. The bacteria don't have histones. No, that would not work because the bacteria, of course, you're right, no nucleus, but they still have DNA and they still have a chromosome. So they would, as far as that goes, they would have histones. But they don't have histones because, again, no differentiation. So for bacteria, whatever you see is what you get. Okay, so actually, both those 2 points, it's the same cause. There's only one cell, so nowhere to export to, and no reason to differentiate because there's just one cell. I'm a little embarrassed. I just spent 20 minutes on 2 lines of this. I'll try to speed it up. But everybody okay so far on this? Basically, just a one-celled organism. No nucleus, of course. I've already commented to you they do not do mitosis because there's no nucleus. Well, they do divide. So it's called binary fission. And that just means that They do double all of the organelles, just like we do, the ribosomes and this kind of thing. Will double. And then half goes here, half goes here, cell divides. But there is a flaw in the system. And the flaw is that this is called asexual reproduction. Asexual reproduction. Now we, of course, and all the animals do sexual reproduction. What is the benefit of sexual reproduction? I know somebody's going to say it's fun. Okay. Yes. But besides that, what is the, pardon me?
Two different sets of DNA.
Go a little further, but you're right. Go a little further with that. Pardon me?
Survival.
No. The two, pardon me?
Disease.
No. Two sets of chromosomes, two sets of DNA. Well, genetic diversity. Genetic diversity. Yeah. So you're right there, but what I'm getting at there, for example, each one of us, of course, is a mixture of our parents, obviously, and a mixture of our grandparents and all the rest of it. We have DNA from each of our ancestors. Well, the bacteria don't do that. So the bacteria, when they divide, there is no genetic diversity. Just each, they're called daughter cells. Each daughter cell is going to have the same DNA as the mother cell. So the mother cell divides, you get two daughter cells, the DNA is identical. Basically, yes?
How does that explain how a bacteria
We're going to cover that at a later time. So hang on for a second, I'll give you a partial answer in a minute, but it's a good point. Where I wanted to go with this is that basically with binary fission, the bacteria are making clones. Everything doubles, there's no mixing, and therefore the DNA of one generation is identical to the DNA of the previous generation. They're all clones. Now the advantage of genetic diversity, not made up by contrast, is
More immune?
that when there's environmental change, like right now, climate change, many of the organisms of a species might die out, but because there is genetic diversity, there will still be enough differences that some members of the species will survive and then carry on for new generations. Well, the bacteria can't do that because they don't have genetic diversity. All the bacteria have the same DNA. So therefore, when there is environmental change, and there's been environmental change before this. There have been ice ages and this kind of thing going on. Of course, never in 30 years like we're doing now, but there's certainly been climate change before this. So as soon as there's climate change, all the bacteria will die. Anything that will kill one bacteria will kill all the bacteria because they all have the same DNA. Now, there's only one flaw in the argument. And the flaw is, it seems to be wrong. Besides that, it's a great argument. Because obviously there are billions of bacteria around. They have not died, despite all of the environmental changes that have occurred before this. So what this implies is, and this now is a partial answer to your question, what this implies is that despite the asexual reproduction, that there is in fact genetic diversity. The bacteria do have genetic diversity. And that's why, sure, many of them will die when the climate changes and stuff, but some will survive. They've already done it. So the question then is, how are they getting this genetic diversity? And that's what I'm going to answer at a later time. But we do it by reproduction. The bacteria do not. They do the genetic diversity by other techniques, not reproduction. So, probably more information than you wanted. Okay, when I give you stuff like this with these numbers, you don't need to remember the numbers. It's just information. So generally, cells will be 0.2 to 2 micrometers long, or diameter, 2 to 8 micrometers long. So bacillus, that would be bacillus, long rod-shaped thing. Coccus would be generally a circle. Again, don't remember the number. It's just information. The shape we've seen before, we've talked about, coccus is spherical, bacillus is rod-shaped, and then spiral or vibrio, it's variable. Curved or corkscrew or something else. Again, my own preference really is just call it miscellaneous. The true answer you'll see in books and stuff, they'll call it vibrio. Lots of these are not curved, so I actually prefer miscellaneous. I won't mark it wrong if you put in vibrio. It's just not very accurate. Okay, so the coccus, we can have diplococcus, so two cocci. By the way, the real